Okay, we're going to talk now about treating for microbial infections. And the idea here is that we want to understand where the weak spots are for the organism and what the ideal characteristics are for an antimicrobial so that we can wipe out the pathogen but hopefully not do too much damage to the patient. So the introduction of modern drugs to control infections was a revolution in the 30s. They've reduced the incidence of infection, but they haven't eradicated infectious disease and never will, and the reason for this is the fact that microbes can mutate and change their characteristics and thus acquire the ability to now escape the antimicrobial effects of a particular drug that was worked up prior to those changes. Doctors are worried that we're close to a post-antibiotic era where drugs we have are no longer effective, and the reason for this is the ability of bacteria, other microbes to resist antimicrobials can be passed from cell to cell to cell. We saw with bacteria 3 different ways of moving DNA around genetic material that can confer new characteristics. This also happens in other types of single-celled organisms as well, through mechanisms such as viral transduction and transposons, just to name a few. The goal of antimicrobial chemotherapy is to administer a drug to an infected person that destroys the infective agent but doesn't harm the host. The drug has to be easy to administer and able to reach the infectious agent anywhere in the body, be toxic to the agent but non-toxic to the host and remain in the body as long as needed, and be safely and easily broken down and excreted. And there are very few drugs that meet all of those criteria. So these are a couple of tables that run through the characteristics of the ideal antimicrobial and some of the terminology that you're going to have to be familiar with when we talk about chemotherapy. I won't go through each of these now, but just kind of star this table and know these terms. Antibiotics are common metabolic products of aerobic bacteria and fungi made to inhibit the growth of competitors in the same habitat so that they can establish their niche in the environment and become dominant. They can be derived from bacteria in the genera Streptomyces and Bacillus, and molds in the genera Penicillium and Cephalosporum, and there are other sources as well in addition to synthetic antimicrobials. Before actual antimicrobial therapy begins, we have to know the nature of the microbe causing the infection, the degree of the microbe's susceptibility to drugs, and the overall medical condition of the patient. The ID of the infectious agent should begin as soon as possible. It should occur before the antimicrobial drugs are given, before their numbers are reduced. Direct exam of samples is a rapid method to detect the pathogen. Doctors often begin therapy on the basis of immediate findings and informed guesses, but statistics also inform the treatment modality. Testing is necessary for staph and Neisseria gonorrhoeae and strep fecalis and aerobic gram-negative intestinal bacteria. Testing for fungal or protozoal infections is difficult and unnecessary. It's unnecessary for organisms known to be susceptible to penicillin G unless the patient is allergic to this antibiotic. The Kirby-Bauer technique involves determining the susceptibility of a pathogen to a particular antibiotic by producing a lawn on nutrient media, and then you hit it with small discs that are impregnated with a selection of antibiotics, and what happens then is you get a zone of inhibited growth around the filter disc, and that the thickness of that zone of inhibition is frequently indicative of how effective that antibiotic is going to be at knocking out that particular microbe. It's not effective for anaerobic, fastidious, or slow-growing bacteria because it's difficult to grow those in culture anyway. And so you can see an example of a typical Kirby-Bauer test. We throw down these little impregnated discs onto the bacterial lawn. You can see what the zone of inhibition looks like. You can measure it with a ruler, and then you can use those findings and compare them to findings in reference material to determine how effective that drug is going to be treating that particular organism. So you can see here a whole host of antibiotics used in a Kirby-Bauer, and the most effective are going to be the ones that have the S by them, okay, susceptible for S. So in this particular block of data, we would go for the chloramphenicol or the bacitracin, or the gentamicin or the kanamycin, okay? And we could also take a look at the vancomycin or the tetracycline. Now, what's going to determine among those drugs that are susceptible what we use on the patient? Well, we had to check out the patient's allergies to those antibiotics by looking at their medical history. And then we have to take into account if anything else is trying to grow in or on that patient that could potentially be pathogenic as well. Tube dilution tests are more sensitive than Kirby-Bauer. The antimicrobial is diluted serially in broth. The tube is inoculated with pure culture. The minimum inhibitory concentration is the smallest concentration of that drug that inhibits the growth. It's useful in determining the effective dose and providing a comparative index against other antimicrobials. Now, why do you do this? Notice the X's, right? The X's mark where we start to see growth inhibition, okay? Now, the point in doing this is that you don't want to use less drug than is necessary to inhibit growth, but you don't want to overuse the drug because there could be negative effects on the patient, or you could end up selecting for other organisms that are pathogenic, that are resistant to that same compound at the higher concentrations. The in vitro activity of a drug is not always correlated with the in vivo effects. Failure of antimicrobial treatment can be due to the inability of the drug to diffuse into that compartment where the pathogen is located. Resistant microbes in the infection that didn't make it into the sample collected for testing, or an infection caused by a community as opposed to a single pathogen. The therapeutic index is the ratio of the dose of the drug that is toxic to humans compared to its minimum effective dose. The smaller the ratio, the higher the potential for drug reactions. For instance, a therapeutic index of 1.1 is very risky. A therapeutic index of 10 is safe. That tells you that the drug is going to knock out the pathogen but have minimal effects on the patient. The drug with the highest therapeutic index has the widest safety margin. The physician has to be careful using the patient's history to inform their treatment before using an antibiotic. Pre-existing conditions can influence the activity of drugs used to treat disease. A history of allergy to certain classes of drugs has to be considered. Underlying liver or kidney disease has to be considered. In individuals that are very old or very young, or those that are pregnant, you have to take special precautions because of an already compromised physiological state. Intake of other drugs can result in a higher degree of toxicity, with failure of one or more drugs possible as well. Also have to take into account genetic or metabolic abnormalities in the patient, the site of infection, the route of administration, and the cost of the drug. Okay, so basically how expensive is this going to be, how effective is this going to be, and is this going to be bad for the patient? You might say, well, how can an antibiotic be bad for the patient? Well, we have to remember, right? Let's take the case of a bacterial infection. We're targeting processes that are unique to bacteria in the design of most antimicrobials, meaning we look at things like how to compromise, say, the bacterial cell wall, or how to compromise the translation apparatus in the bacteria based on the ability of these antimicrobials to interact just with the bacterial machinery and to leave our machinery alone. But you have to recall, okay, number one, that there is, in a sense, bacterial machinery inside every single one of our cells if we consider mitochondrial activity, right? Because we think mitochondria are descended from bacteria. A high enough dose of an antibiotic could potentially inhibit mitochondrial function. Another consideration is the ability of the liver and the kidneys to clear this stuff so that it doesn't build up to toxic levels in the interstitial fluid and thus kill cells and tissues. The liver and the kidneys are tasked with clearing toxins from the body, and if we overload those organs, not only can they fail to clear the antibiotic, but they might be unable to clear other toxins as well. An example, we could have an elderly alcoholic with pneumonia caused by Klebsiella, complicated by diminished liver and kidney function, requiring parenteral drug administration, that means other than through the GI, and a history of allergy to penicillin, and a cancer patient with severe systemic candida. Even when all the information is in, the final choice of the drug isn't easy or straightforward, right? Now think about these two cases for a second, right? Elderly alcoholic with pneumonia caused by Klebsiella, complicated by diminished liver and kidney function, meaning what? The ability to get these drugs out of the system is compromised in this patient, and so we're going to have to find the minimum inhibitory dose, and it can't be in a class of drugs related to penicillin, right? Because he's already shown a hypersensitivity. In a cancer patient, we have reduced ability to deal with our normal flora and fauna, let alone our pathogens. The cancer patient is already immunocompromised as a result of the chemotherapy or the radiation therapy they're undergoing. And so if we hit them with an antibiotic, then it may be difficult for their body systems to clear that component out before it creates toxic conditions. Okay, take a look at selective toxicity, ID some major targets of antimicrobials, and talk about kind of how these drugs work and what they're effective against and the mode of action. We'll explain how drugs that target folate synthesis work. We'll talk about fluoroquinolone. We'll describe the mode of action of drugs that target the plasma membrane and those that target the cell wall. We'll name categories of antifungals and antiprotozoals and antihelminthics, and we'll list major targets of antivirals. So, the goal of antimicrobials is to disrupt cell processes or structures of bacteria, fungi, or protozoa, or to inhibit viral replication, or to interfere with the function of enzymes required to synthesize or assemble macromolecules in that pathogen. It destroys structures that are already formed in the cell. It is usually selectively toxic, meaning it kills or inhibits the actions or synthesis of molecules and microbes, but not vertebrate cells. So the idea here is we go after the bad actor and we leave our own machinery alone. Drugs with excellent selective toxicity block the synthesis of cell walls because that's unique to bacteria. We don't have anything like that in our system. Drugs most toxic to humans act on structures common to both the infective agent and the host cell, such as the cell membrane. As the characteristics of the infection agent are more and more similar to our own characteristics, selective toxicity gets tougher to pull off. And that's why it's tough to make a good antiviral, and why it's tough to make a good antiprotozoal or antihelminthic, because there aren't enough unique targets for the drugs to do their tricks. The goals of chemotherapy are to ID the structural and metabolic needs of the living cell and removing, disrupting, or interfering with those requirements so that the pathogen dies. Antimicrobial drug categories can inhibit cell wall synthesis, inhibit nucleic acid synthesis and function, inhibit protein synthesis at the level of translation, interfere with the assembly of the cell membrane, or inhibit folic acid synthesis. In your book, Table 10.4 lists the targets for a variety of antimicrobials, and you can see here an example of a few of the different antimicrobial drugs. Okay, so let's just take a mini tour through here. All right, so here we have at the top left, we'll do this in red, action against ribosomes, right? These guys attack the 50S subunit. These guys attack the 30S subunit of the ribosome. The ribosome is the protein building machinery inside both bacterial cells and eukaryotic cells. We're made of eukaryotic cells, not bacterial cells. So the idea is with this family of drugs here, okay, and even these guys that hit both, you're only going to hit bacterial translation and not our own translation in the cytoplasm of our cells that takes place on the rough ER or in the cell cytoplasm. Now, up to a certain point, right? Because remember, these could affect the function of mitochondrial ribosomes. Mitochondrial ribosomes. Okay, at high enough concentration. So that's our therapeutic window, right? Hit the bacterial translation, leave the mitochondria alone. Okay, folic acid synthesis. Okay, folic acid is critical in a lot of pathways that involve the production of metabolic energy. Sulfonamides are examples of antimicrobials that block the production of folic acid, meaning that the only way that the microbe can get it is to pull it out of the environment. Okay, so it's like you're taking away a critical part of the structure of the enzymatic pathway so that it can't work anymore. Okay, now you might say, okay, well, what about our cells? What about our folate? Well, how do we get our folate? We eat it. Okay, it's in our diet. These guys have to make it in large part in order to function. And they have unique enzymes that we don't have that produce the folate. And so if we block those enzymes, we block the pathogen and we don't really affect our own cell function very much. In fact, we get some of our folate from bacterial activity in our colon, right? The E. coli that live there whip out folate for us. So a possible bad side effect of this is that you could knock out some of our normal flora, okay, but that can always grow back later. Cell wall inhibitors, okay, there are no cell walls in eukaryotic cells in our body, so all of these guys are selectively toxic. Okay, so you might think, okay, can I just take infinite amounts of this at any high enough concentration in order to wipe it out? Well, no, okay, because again, the liver and the kidneys have to clear this stuff. And you want to have enough of this to knock back the pathogen, but you don't want to have so much that you end up damaging your liver, you know, promoting things like hepatitis or throwing you into kidney failure. Okay, cytoplasmic membrane. Okay, so these guys are going to interfere with phospholipid function. Now, this is getting pretty close to home because we have phospholipids in our own cells, so we have to be careful with how we use these. The therapeutic index for this is going to be a lot lower than this group or this group over here. Okay, now these guys, the DNA and the RNA, can also be specifically inhibited in the prokaryotes because while the enzymes that produce DNA and RNA in the prokaryotes are functionally similar, they are structurally distinct. And again, where's the danger here? Where's what can we end up with a high amount of drug here? We could inhibit mitochondrial DNA and RNA synthesis, right? It would have to be pretty high to do that, but again, that tells you why you can't just go infinitely high on these drugs. In addition, with all of these, you could adversely affect liver and kidney function. Broad-spec drugs are effective against more than one group of bacteria, such as tetracycline antibiotics, while narrow-spec hit a specific group, like polymyxins. Penicillins attack cell wall synthesis. It was initially a narrow-spec that was discovered as a byproduct of mold growth. It's been improved over the years so that it's more stable and more effective. Later penicillins overcame the limitations of the initial discovery. Carbenicillin is an example. Methicillin is another one. More stable versions of penicillin. But you may have heard of well, we're on the topic of penicillin, a condition called MRSA. Okay? MRSA. MRSA. That's an acronym, stands for Methicillin Resistant Staph aureus. Well, what's methicillin? Methicillin is a cousin of penicillin. Okay, it's a derivative. And Staph aureus, in a lot of clinical settings, has evolved to be resistant to this drug, and it's one of the reasons why it's a major source of nosocomial infection. And so here you can see some of the different spectrum of activity for the antibiotics. I won't go through each one of these, but this is 10.5 in your book. It's rare that bacteria are all on their lonesome. They generally work in communities, either with similar bacteria or identical bacteria or other microbes. And the result of this is that as they act as a community, they're more resistant to antibiotics than if they try and simply function on their own. So they behave differently when they're free living than when they're in communities. They're often unaffected by antimicrobials. Antibiotics often can't penetrate the extracellular material in a biofilm, and so we can't get in there and get at the pathogen effectively. Bacterial biofilms express different phenotypes and have different antibiotic susceptibilities than free-living bacteria. Biofilm treatment strategies include interrupting quorum sensing, which is what? This ability of the community to change its physiology based on the change in environment that it picks up on. Daptomycin has shown success doing this. Adding DNases to antibiotics can aid in penetration through extracellular debris, because one of the major extracellular waste products when the bacterial cell dies is tons and tons of DNA. Impregnating devices with antibiotics prior to implantation is another effective method, another approach. Some antibiotics cause biofilms to form at a higher rate than they normally would. So that's what's called an unintended consequence. Now, fungal infection is a different animal. Okay, eukaryotic cells present special problems in chemotherapy because they are a lot like our own. Drugs designed to act on bacteria are ineffective against fungi because they're a different kingdom. Okay, they have cell walls, but they're made of chitin. They're eukaryotic. They live off of dead stuff mostly. Okay, the similarities between fungal and human cells mean that drugs toxic to fungi can harm us a lot more quickly. Only a few agents with special antifungals have been developed, and you can see them here. Okay. Okay. When last we left off, we were talking about agents that are used to control microbes, and when we're discussing protozoa, we have a unique challenge, which is that protozoans are eukaryotic, and their cell structure thus is very similar to our own cell structure. So it's difficult to generate a drug that's going to specifically impede the life cycle of a protozoan while not affecting our own cells and tissues. Quinine has been the principal treatment of malaria for hundreds of years. It's been replaced by quinolones, chloroquine, and primaquine, which are synthetic derivatives. A lot of species of Plasmodium and many stages within its life cycle means that no drug is universally effective. The life cycle is relatively complex when you're looking at protozoans. They have some stages in which they form a cyst-like structure that can persist inside living tissue and is relatively resistant to the penetration of antiprotozoals, and then the free living stages where they tend to be more vulnerable. Metronidazole is widely used as an amebicide. It treats intestinal infections and hepatic disease caused by Entamoeba histolytica, and this is the agent of amoebic dysentery. It also treats Giardia lamblia and Trichomonas vaginalis. Other drugs with antiprotozoal activity include quinacrine, sulfonamides, and tetracyclines. Antihelminthics face the same challenges as antiprotozoals. Flukes, tapeworms, and roundworms are larger parasites, and their physiology is even more similar to humans than single-celled protozoans. Blocking reproduction does not affect adult worms. The most effective drugs are going to immobilize, disintegrate, or inhibit the metabolism of all stages of the life cycle. Mebendazole, albendazole are both known to inhibit microtubule formation in worms, eggs, and larvae. Pyrantel paralyzes the muscles of intestinal roundworms. Praziquantel is effective against tapeworm and fluke infections, and ivermectin is a veterinary drug used for strong lordosis and onchocirrhosis in humans. Now, ivermectin is unique. It was developed as the result of a soil sample survey conducted for antifungals, and it turned out that its accidental isolation demonstrated in the presence of protozoans that it was an effective ecto and ento antiparasitic. Viral infections are even more difficult to treat because, as we know, viruses are utilizing the machinery of the host cell in order to reproduce. The infectious agent relies on the host in order for it to make new viral particles. Disrupting metabolism requires disruption of cellular metabolism or the isolation of a unique viral target, and there aren't that many unique viral targets. Measles, mumps, and hepatitis are prevented through vaccines, and this is a different method of avoiding infection via virus, because in a vaccine what you do is you prompt the immune system for the presence of a particular antigen, which is on the surface of the pathogen you're trying to prevent. That's totally different than trying to deal with an infection that's already been established by a virus. AIDS, flu, and the common cold attest to the need for more effective meds in the treatment of viral medication. And again, viruses, since they rely on the host cell machinery to reproduce, A, don't provide a lot of unique targets, but B, they can compensate for or deal with a higher mutation rate. As a result, they're more prone to changing their antigenic display and thus avoiding the immune system than technically living pathogens are. Major modes of action of antivirals include barring penetration of the virus into the host cell, blocking transcription and translation of viral molecules, preventing the maturation of viral particles. Double-stranded RNA-activated caspase oligomerizer is a breakthrough antiviral treatment, what it does is it looks for double-stranded RNA, which is a unique molecule in viral replication, and destroys it. And as a result, you can't assemble an effective viral particle. Other examples include reverse transcriptase inhibitors such as AZT, which target an enzyme unique to the virus. So here you can see some of the modes of actions of antivirals. I won't go through each one. Just note that we want to be aware of how they work. Okay. We'll talk about 2 ways that microbes acquire antimicrobial resistance. List 5 cellular or structural mechanisms that microbes use to resist antimicrobials, and talk about at least 2 novel antimicrobial strategies that we're currently undertaking. So drug resistance is an adaptive response in which microbes begin to tolerate an amount of drug that would normally inhibit their growth. Due to the genetic versatility and adaptability of microbial populations, this characteristic can be intrinsic, meaning it's already within the genetic material, or it can be acquired as a result of moving DNA between microbes. And we've already seen some of the mechanisms whereby that happens: transfection, transduction, transformation, just to name a few. Resistance to penicillin developed in some bacteria as early as the 40s. In the 80s and 90s, scientists began to observe treatment failures on a large scale. Microbes were becoming newly resistant to a drug after one of the following: spontaneous mutations in critical chromosomal genes, and the acquisition of new genes or sets of genes as a result of gene transfer, either within or between species. Chromosomal drug resistance usually results from spontaneous random mutation. Changes in drug sensitivity can be overcome with larger doses of drug or with the development of drug derivatives. Persisters slow or stop metabolism so that the microbe can be harmed by the antibiotic. Resistance to horizontal transfer can occur through conjugation, transformation, or transduction. Remember that mobile genetic elements in bacteria called plasmids often host antibiotic genes, and thus, when passed from one cell to the other, confer that characteristic on the recipient. Plasmids that have these characteristics are often present in microbes before they've been exposed to the antibiotic, and that's how natural selection works rapidly in a bacterial population. Transposons can duplicate and insert genes for drug resistance into plasmids, and the sharing of resistance genes via conjugation and other methods of transformation accounts for the rapid proliferation of these drug-resistant species. And so you can see some of the different mechanisms for drug resistance here. We can target the translation machinery of the bacteria, because while it's functionally similar to eukaryotic translational machinery, the tools are slightly different. We can, as a result of using such a drug, select for strains resistant to the interaction of the drug with the translational machinery. Enzymes can be synthesized that basically destroy the active component of the drug. For instance, shown in the top there, penicillinase can hydrolyze the lactone linkage that turns the molecule from an active to an inactive form, and thus it's no longer effective at compromising the structure of the cell wall. We can have changes in permeability so that the drug can't penetrate, and we can have the drug eliminated from the bacteria through a novel membrane pump or channel, so that the drug is shot out of the living cell before it gets a chance to prove its toxicity. Development of resistance has consequences. Any large population of microbes has a few cells that harbor drug resistance. as long as the drug isn't present in the habitat, the genes are not expressed. If the drug population is exposed to drugs, the drug-resistant population has a selective advantage, and its offspring will inherit that characteristic. As a result, the replacement population isn't going to be susceptible to the same treatment, and you'll have to come up with another way of dealing with infection by this selected population. The threat report from the CDC in 2013 outlined a catastrophic antibiotic resistance situation. We may be entering a post-antibiotic era where some infections can only be treated through either vaccination or through natural destruction of the pathogen through action of the immune system. New and effective antibiotics have been slow to come to market. Antibiotics aren't economically lucrative, and they're time-consuming and expensive to work up. Urgent threats include C. diff and carbapenem-resistant enterobacteria, drug-resistant Neisseria gonorrhoeae. Other threats include multi-drug-resistant Acinetobacter and drug-resistant Campylobacter, fluconazole-resistant Candida, that's a type of fungus. Concerning threats include vancomycin-resistant staph, erythromycin-resistant group A strep, and clindamycin-resistant group B strep. So what are some novel approaches that we can put into effect in order to generate a new type of antibiotic that has yet to show the ability to be neutralized as a result of resistance gene? We can target the iron scavenging capability of bacteria, or use RNA interference, where small pieces of RNA can turn down genes that are critical for drug resistance and thus shutting down the metabolism of the pathogen. Drug trials have also been undertaken to evaluate the effectiveness of synthetic RNA in treating Hep C and respiratory syncytial virus. We can mimic defense peptide molecules. Peptides of between 20 and 50 amino acids are secreted as part of mammalian innate immunity. Things like defensins, meganins, and protindigrins have been known long before the extensive characterization of our adaptive immunity. Bacteria also produce defense peptides called bactericidins and antibiotics in order to carve out their own niche within their ecosystem. They insert into membranes and target other structures in the cells. They may be more effective than narrowly targeted drugs and less likely to foster resistance. We can use bacteriophage. Eastern European countries use mixes of bacteriophage as medicine, but these drugs have never been approved for use in the West. Biophage PA has been used to treat ear infections caused by Pseudomonas aeruginosa biofilm. Other researchers are incorporating bacteriophage into wound dressing. Advantages to phage treatment is their narrow specificity. They only hit one species of bacteria. We can also use probiotics, which are preps of live microbes that you can ingest and thus crowd out resources for the pathogen. They can replace microbes lost during antimicrobial therapy as well. They're safe and effective. They're useful in the management of food allergies. Prebiotics are nutrients that encourage the growth of beneficial microbes. Fructans encourage the growth of bifidobacterium in the large intestine and discourage pathogenic growth. Fecal transplants have been used to treat C. diff in ulcerative colitis. The transfer of feces from a healthy patient through colonoscopy is another approach. Work is also underway to develop a pill that contains the species that can recolonize the colon. Okay, in this section we're going to distinguish between drug toxicity and allergic reactions to drugs and explain a superinfection and how it occurs. So drugs administered to the host, even though its target is the pathogen, and the effect of the drug on the host has to be taken into account for effective treatment. Ideally, you want the drug only to hit the pathogen, but there's going to be effects on the host organism, on the human being. 5% of all persons taking an antimicrobial will experience an adverse event in any large population. They can affect the liver, the kidneys, the GI, the cardiovascular, the nervous system, the respiratory system, the function of the skin, as well as the bones and teeth. And you can see what some of the effects are here in Table 10.10. An allergy is simply an overzealous response of our innate immunity to the compound or the entity that's being considered. Okay, in this case, we're talking about a drug that's meant to alleviate the effects of an infection, but the drug itself can contain surface features that act as allergens. And the result is that the immune system, in a misguided attempt to defend you against this compound that's actually meant to help you, produces physiological responses that are uncomfortable. It can be provoked by the intact molecule or by metabolic breakdown products of the drug. They've been reported for every type of drug, but penicillin allergies are one of the most common. Sensitization occurs during first contact with the drug. A subsequent exposure leads to hives, inflammation, or anaphylaxis. Normal microbial colonists of the healthy body surface are protective in that they utilize important compounds of that environment that become unavailable to the pathogen due to competition. In other words, we're here first, we take up the nutrients, we take up the space for reproduction, and we crowd out the potential pathogen. These are beneficial bacteria, or at the very worst, neutral to the function of the body. But there are some that are pathogens that are kept in check by these larger populations. Broad-spec antimicrobials can wipe out both the pathogen and these useful microbes and can give the pathogen a chance to become the dominant component of the population during the recovery process. So what happens in a superinfection is that microbes that were once a small number of that population overgrow when the resident biota get knocked back by these broad specs, and the result is an infection. A UTI can be caused by E. coli treated with antibiotics. Lactobacillus in the female vagina get wiped out by broad-spec cephalosporins and other drugs of the same class used to treat UTIs, and then you get overgrowth of yeast causing an infection or oral thrush, which is whitened patches on the mucous membranes indicating that the fungus is beginning to take hold. Antibiotic-assisted colitis, it can occur as a result of oral therapy with tetracycline, clindamycin, or other broad specs that kill off the normal biota of the colon. Resulting overgrowth of Clostridium difficile, or what we sometimes refer to as C. diff, invades the intestinal lining, releasing toxins, producing diarrhea, fever, and abdominal pain. Worldwide problems in the management of antimicrobial drugs is creating a challenge that may not be overcome as a result of research into new therapies designed to control pathogenic growth. Antimicrobials were once seen as a cure-all for infections like the common cold and acne, and in many cases these drugs have no effect or are actually harmful. About 200 million prescriptions for antimicrobials are written in the U.S. annually. 75% of these are for pharyngeal, sinus, lung, and upper respiratory infections that are viral. And so you might wonder, why is the physician using an antimicrobial to treat a viral condition? And that's usually to avoid this idea of what's called a secondary infection. So, secondary infections result from the weakened condition of the tissue following pathogenic infection from something other than a bacteria. And so the drugs are given prophylactically to avoid this pathogen moving in behind, say, a viral infection, in the hope that the secondary infection never sets up. The downfall of this, though, is that it can act to select for resistant strains of microbes to that antibiotic. So this has led to superinfections and adverse reactions. They can be caused by the development of resistance in bystander microbes that are exposed to the drug, leading to the selection for resistant pathogens. This awareness has led to the reduction in the universal use of antibiotics in situations where they're not warranted. Lots of excess antimicrobials in the United States are exported to countries where controls aren't as strict. It's common for individuals in Latin America and Asia to self-medicate with antibiotics. The drugs are used in this way become largely ineffective and leading to drug resistance due to selection. Every allied health professional needs to be aware not only of the admirable but also the utilitarian nature of these classes of drugs and their limitations. Again, when you limit the use of these things, they're going to be more effective because you're less likely to select for resistant microbes within the population. Okay, that brings us to the end of this chapter. Again, review this material as it will be covered on the upcoming quizzes and exams, and I'll see everybody in the next podcast. I thank you for listening.